Hi, guys. Happy Tuesday. It is Mrs. England recording from normal, and it is Monday, and I'm recording the video for you for tomorrow. Um, yeah, this is how I'm feeling today. Just a little bit like bleh, but it's another week. Um, I really have been enjoying your flip grids. So if you haven't had a chance to get that done, please do that. Just want to see your faces. Make sure you're doing okay, and everybody else wants to see you too. Plus, like I said, it's awesome to see you guys in your natural habitats. Okay, today, which is Tuesday, um, my goal is to review what you did yesterday and kind of like review the whole story so that we're almost to the end of understanding all of this. So we're going to review what we've done so far. And then as you can see on number 2 here, there is no new content today. We are just going to apply what we did yesterday. So yesterday you were really just kind of coming familiar with the terms of reproductive isolation. And today we're going to look at some real world data and apply it. So your assignment will be to do the Salamander lab. You're welcome to do it with a partner. Um, and just each of you fill out your own sheet. There is a Google drawing that goes along with it that you'll be looking at data on, and there's the lab questions 1 through 10. Okay, so let's start with review. This unit is on evolution. Evolution, as we know, is the heritable change in populations over time. So when we look at the fossil record, we know that life has not remained the same over time. So how does it change? Well, one way it can change is through natural selection. This is a description of how populations of organisms change over time. And when they change over time due to natural selection, we notice that organisms are more well suited to their environment. So we looked at our frogs. So this may not be your picture, but it's similar. Here is evolution. So you started with the brown frog, and over time, you ended up with a brown frog with blue legs. This kind of change is called microevolution, where the population just shifts to be slightly different color. And then where the population splits into 2 different species, that's macroevolution. So they both have the same how. Um, it's just what the results are that's different between macro and microevolution. So both of those processes happen through natural selection. And in natural selection, we know that you have to have variety in your population, that the variety must be heritable, and then the variations that you have, somebody must have differential reproduction or reproductive advantage. So over here, we can see evolution because the original population is mixed and the final population is almost all brown, looking much more well suited to its environment. So we would say that these beetles now look adapted to their environment. They have a brown adaptation. And how did they get that? As a result of having a reproductive advantage. So the end result of natural selection is adaptation, or populations appearing to be well suited to their environment. Then we went into the 3 types of natural selection. There's directional, where it just shifts to be bigger, smaller, darker, lighter, taller, shorter. Stabilizing, where your intermediate is selected for, and then disruptive, where you have 2 different phenotypes selected for at the same time. When we look a little bit more closely at disruptive selection, where your population becomes separated, could lead to gene flow stopping. So if gene flow stops between this population and this population over here, that can result in mutations adding up between them, so much so that when they come back together, if they ever do, they don't mate with each other. So disruptive selection could result in speciation. If there's any of reproductive isolation, which we learned yesterday, doesn't just have to be geographically, like you're separated by a mountain, but it can be behaviorally, like you have different mating rituals, you have different mating calls, mating dances. You could reproduce at different times of the year, like the frogs, with some reproduce in the early spring and some reproduce in the late spring. You can have mechanical separation where your reproductive parts no longer go together. Or you could have some sort of post-zygotic where you actually reproduce, but your offspring don't survive. So it is this process that leads to new species on Earth and explains why there's so much diversity. Okay, so if we want to look at an example applying yesterday, we would look again at an example we've looked at multiple times. We would look at polar bears and grizzly bears. Why do scientists consider these a different species? Well, they had geographic separation, which in those different environments, they have different adaptations. So obviously a polar bear has gotten multiple mutations that has made it be white, have big claws, have the ability to hunt on ice, whereas grizzly bears, they have more of a variety of diets. They have the brown fur, they have a smaller size. Now, interestingly, they do come back together today, which is kind of rare, but if we look at what's happening to the polar bears environment due to global warming, and then if we look at what's happening to the grizzly bears environment due to deforestation, there's actually less habitat pushing them back together, and they do have offspring. There is evidence of in nature growler bears or pizzly bears, whichever way you want to call it. However, we would say this is post-zygotic hybrid inviability because this guy isn't very good in either environment. He doesn't have good camouflage in the Arctic, and he doesn't have very good camouflage in the grizzly bear's habitat, like in Alaska. So probably this guy's going to have a reproductive disadvantage. So this is another example. They do mate, but when they mate, their offspring is not going to survive very well. So you're selected against if you mate with someone who does not look like you. So polar bears have a higher advantage if they mate with someone who looks like them, as well as grizzly bears. Okay. So, just to be clear, where do new species come from? Where did these two new species come from? They evolved from an existing population. So species arise from existing populations. Just new variations in existing populations give rise to the diversity we see in life. So life only comes from pre-existing life. Okay, that's the end of the review. Today's lab, we're going to look at a population of salamanders, okay? And we're going to ask the question, are these salamanders going through speciation? Okay, the science skills that you're going to be working with today, exercising those brilliant brains of yours, is you're going to analyze and interpret data. So you're going to get some scientist data. And then you're going to use that data and all of the background knowledge you have that we just went through to construct an explanation for how speciation is going to occur. Okay, salamanders. Let's look at this guy. Do you know what this organism is? I wouldn't have. A lot of students will call it a lizard. It is not a lizard. It is, in fact, our Illinois state salamander. So let's get a little introduction to salamanders. Now, in this video, this is not our eastern tiger salamander. This is the barred salamander. He's a little different, but at least gives you a good intro and a visualization of our salamanders. So let me play this quick video for us.
With a face and mouth like a muppet, some might think this barred tiger salamander is cute, but to insect passers-by, that mouth is deadly. This salamander, with enough camouflage markings on its body to almost blend into the surrounding foliage to an unsuspecting passing insect, can quietly stalk and wait for the moment. With a quick opening of the mouth, it captures its prey. And with another quick opening and gulp, it then swallows its victims. Even though they can grow to over a foot long and are the largest land-dwelling salamanders in the world, they are hard to find. And they are mostly active at night, when it's harder for us to see them. These amphibians only have 4 toes on their front feet and 5 toes on their back feet. And they can live up to 20 years. Besides insects and earthworms, these salamanders can also eat small mice and smaller amphibians like frogs. They even cut down on some of the nighttime noise by chomping down on a cricket every now and then. They live in places where they can stay cool and moist, and they require water to lay their eggs. They spend a lot of their time in underground burrows, sometimes borrowing the homes of crayfish or even prairie dogs. Tiger salamanders can be found throughout North America, but the barred tiger salamander is only found in a smaller range from Nebraska to northeastern Mexico. And in Kansas, they are the official state amphibian.
Okay, so salamanders are amphibians. They do go through metamorphosis. So this is what their metamorphosis. They lay eggs. They spend time as gilled larvae. So some of them retain their gills even after they have matured, like the axolotl, which is only found in Mexico. It's a really cool one. He's white. You might want to look him up. But then the ones we're looking at actually, after they have emerged from their eggs and they're larvae, they go through a metamorphosis and they'll become terrestrial. Okay, so these salamanders that we're going to look at are these species here, or subspecies. Okay, so why are we looking at California? Well, again, because that's the data that's provided for us. If I had data on Illinois salamanders, that would be fabulous, but I don't have any real data, and I love it when you guys look at real data. Okay, so I'm going to play the intro video and you will see some of our salamanders. So here is the professor that has shared his data with us. And we're going to just watch about 20 seconds of this video to see, for him to kind of set us up with his data.
Meet a type of terrestrial salamander found in California. Now meet another. And another. These salamanders are members of the same species, so why do they look and behave so differently?
Okay, so those are our cute little salamanders that we're going to be looking with on this lab. Now I'm going to click over to the lab. This is on the classroom for you. This is your assignment. All right, so you're going to start up here. Our lab question is, are the two populations of salamanders in California separate species? Okay, and here's a little background question. So what do you know? What is this guy? I hope you know it's a salamander. What are some things you learned about him from my pre-lab? So think about what you learned about salamanders that you didn't know, maybe what they eat or where they live. And go ahead and answer that question. Our lab goal says in class, we've been looking at examples and deciding if two species are the same species or if they're separate species. Today, you will look at a real life population of salamanders living in California. You will look for patterns in their adaptations and in the hybrid and construct an explanation as to whether the two populations are the same species or separate species. So today, you're going to do the analysis and just the lab questions. Tomorrow, you're going to do the model. So you're going to do a visual and a written explanation of what you figured out here. There is 3 different claims that you could make at the end of this data analysis. You could claim, number 1, the salamanders of California are variations of the same species, so they're just straight up the same species. So you could claim that the salamanders of California are in the process of becoming different species, or you could claim that the salamanders of California, that nope, they are indeed different species. What do you think the data tells you? Okay, so here's how it works. The lab background. The small salamanders are in the genus Insatina, and they are strictly terrestrial. They even lay their eggs on land. Nevertheless, these salamanders need a moist environment and do not thrive in the arid regions. So this is based upon the work of Professor Stebbins. Okay, and here's where the procedure starts. So let me walk you through this a little bit. Okay, so if we're in class, what you would have is you would have this map of California. Okay, and in this map of California, what I wouldn't have done for you is I would not have colored all the squares for you. So you would have been given the GPS coordinates and you would have colored in all of these boxes, what these boxes represent is where different salamanders were found. All of these are the different subspecies. So these are all the same species. And what they did is they plotted where they found salamanders in California and what they looked like. Okay, so a little bit more on the background of California. Let me scroll down to right here. Okay, so in California, this area here is the Great Valley that is deserts. Okay, so there we didn't find any salamanders in that area, and you'll see that on our grid that we did. And over here to the east of the Great Valley are the Sierra Nevada mountain ranges, and they are some of the places that we found salamanders. And then we also found salamanders in the coastal mountain ranges, which look a little bit like this. Okay, so when we click over here in our lab, you can see that there are colored squares both to the east, where the Sierra Nevada mountain ranges are, and to the west, where the coastal mountain ranges are. But there are no salamanders where all these gray X's are, and that is due to the fact that that is a desert region. Okay, and the ancestral population of salamanders actually started up here. And what happened is as the population increased, so up here is the redwood forest, some of you have maybe been there before, their population started there. And as they grew in population, they actually migrated south, and some migrated on the route of the Sierra Nevadas, and some migrated on the route of the coastal mountain ranges, but they avoided the Central Valley. And then down in these areas, the two populations actually come back together. So we're going to see what happens when these two populations come back together. This is like a real world application to what you guys did with your frogs. Okay, so what you guys need to do is you're going to double click on this Google drawing for the map of California. You're going to find the colors in parentheses on these cards. So I'm going to go ahead and demonstrate for you. Okay, so here's the subspecies name. This is how many were found, and this is where they were found. So what I'm going to do is I'm going to move this over here. Oops, is it not going to let me? This picture over here to where the purple squares are. And I'm just going to lay him right there. Okay, I'm going to do that for all of them. The brown, I'm going to pull down here. Try not to put your pictures. Tip is try not to put your pictures in the Great Valley, but put your pictures like I've done to the left or to the right of the Great Valley. It will help you when you're going to analyze your data. So all you do is you pull your pictures. So this brown one right here would go down here by the brown. The orange ones go by the orange. There are a few colors that don't have a picture for it. That's okay. You'll see about those in the questions. Okay. I'm going to click Save and Close. So that's what the instructions say. Double click on the map of California, find the color in parentheses, match the color in parentheses to the color squares on the map. Draw the picture close to the colored squares. So drop, that should say drop. Oops, let me fix that before I drop. The pictures close to the colored squares. Do not put any pictures in the valley. Recall from the pre-lab, the valley is arid or dry, so no salamanders were found there. When done dragging pictures, look for patterns that emerge in the coloration of the salamanders. Okay, so that goes pretty fast because I've done all the plotting for you. All you have to do is match the pictures to the area that they were found in. Then you're going to come down here and you're going to answer questions 1 through 10. And that marks the end of today's activity. All right, guys, I hope you're doing well. Email me if you have any questions. Many of these questions will be hypotheses, just your best guess. You may not know yet. We'll talk about the answers to these tomorrow, and hopefully you have enough information tomorrow to put together your, on Wednesday, and to put together your final model. All right, guys, bye for now. Have a great Tuesday.